Welcome to CFRI's Cystic Fibrosis Community Voices, a video podcast series created by and for the cystic fibrosis community.
Hello, and welcome to CFRI's CF Community Voices, coming to you from our home studio in Palo Alto, California. I'm Sue Landgraf, Executive Director of Cystic Fibrosis Research Incorporated, CFRI, and I'm very pleased to welcome our viewers and listeners. We have a very special program ahead, but before getting started, I want to thank our generous sponsors, Vertex Pharmaceuticals, Chiesa USA, Gilead Sciences, Genentech, and Proteus Therapeutics. I also would like to note that no information presented in this podcast is intended for a patient's diagnosis or treatment. We hope you enjoyed the podcast. Thank you, and let's get started.
Hello, and thanks for inviting me to be on your podcast. It's an exciting opportunity to talk to a different group of people than I normally talk to about the research we're doing in the lab and why I've gotten interested in applying novel technologies to cystic fibrosis. So my name is Matthew Porteus. I'm an MD PhD. I was trained at Stanford and then ended up getting my training in pediatric hematology and now run a research lab at Stanford, and I practice medicine at Lucile Packard Children's Hospital. I first learned about cystic fibrosis in medical school as we were learning about genetic diseases, which of course is a little bit sterile because it's in a book. And then my first patients with cystic fibrosis as I was rotating through pediatrics as part of my medical school rotations. But even in that situation, I have to admit that my direct interaction was still a little bit at a distance, and I really first became engaged in taking care of patients with cystic fibrosis in my pediatric residency at Boston Children's Hospital, which has a very large cystic fibrosis population, and really, every day I would go in and talk to the patients who were in the hospital, usually 2 or 3 of them at any given time, about how they were doing. And some of these patients were young adults, some were still children, and they really taught me a lot about their disease, about what they were going through, about life living with cystic fibrosis. I ended up deciding to go into pediatric hematology because I was interested in another genetic disease called sickle cell anemia, which affects the red blood cells. But they're very similar in that they're genetic diseases that people are born with. And they're very similar in the sense that in the current day, with improved therapies, people are living through their childhood, which once didn't happen, but they're still live lives that are full of challenges and still characterized by people dying before they should. And because these are genetic diseases, and by that I mean that people are born with specific changes in their DNA in a single gene. So for sickle cell anemia, that's a gene called beta globin. For cystic fibrosis, of course, that's the cystic fibrosis transmembrane conductance regulator, or CFTR gene. That means they have the disease from birth. Now, it doesn't always show up right at, sometimes it shows up right at birth, sometimes the manifestations show up later. And so what I became interested in as I was learning about taking care of patients and learning about cystic fibrosis and sickle cell anemia was the idea that if this is a genetic disease caused by changes in a gene, why couldn't we reverse that gene so that it didn't cause the disease? And I have to admit that when I talked to some of my mentors about this, they were very supportive, but they also pointed out to me that this was going to be a very challenging and probably lifelong endeavor. What's been exciting over the last 15 years, but really what's accelerated over the last 3 or 4 years, is that the ability to change the DNA sequence of a cell, in particular stem cells, has accelerated. And so that process is what we call genome editing, because like the way you edit a document on your computer and change a single letter to another letter, we now have the capability of changing single letters in the DNA code of cells so that we can change letters that cause disease into letters that don't cause disease. So the general concept for sickle cell anemia, which is a genetic disease of the blood, is to take stem cells of the blood, which are stem cells that don't make lung, they only make blood cells, and change the DNA that causes sickle cell disease to the DNA that doesn't. And in the laboratory now, we can change the stem cells of patients who have sickle cell disease. We can fix that mutation at a frequency of around 60% of the time, which is a level of correction that we think will be able to cure these patients of their disease. And so what we're doing in the lab now is taking that process that we do in the lab and moving it into a specialized facility and then hopefully into clinical trials over the next 12 to 18 months. So about a year and a half ago, maybe 2 years ago, as we were getting very good at doing this genome editing in blood stem cells, some collaborators here at Stanford and actually a friend of mine from when I was in medical school and a postdoctoral scientist who wanted to join my lab started talking about, well, if we can do this for sickle cell disease, why can't we do this for cystic fibrosis? And so we started thinking about, well, what have we learned for sickle cell disease and how might we be able to apply that to cystic fibrosis? Because the concept, as I was describing before, is quite similar. The idea would be to take a patient's cells and change the sequence so that the cells no longer have the mutation, so we would fix the CFTR gene. But there's some really big differences between the two diseases as well. Probably the biggest difference is that for diseases of the blood, like sickle cell anemia, we know how to remove the stem cells out of the body. We then just had to figure out a way of correcting the stem cells, and then we know how to give the stem cells back. The challenge with cystic fibrosis is, of course, it's not a disease that we know how to remove stem cells from any of the organs that are affected by the disease. So we don't really know how to remove stem cells from the lung and give them back. We don't really know how to remove stem cells of the intestine and give them back. We don't know how to remove stem cells from the pancreas and give them back. And so we started thinking about, well, just because we don't know how to do it doesn't mean we shouldn't start working on it. And so again, about a year and a half ago, again, talking to some of my colleagues here at Stanford, Dr. Kuo, who's an adult hematologist and who is working on lung stem cells, Dr. Desai, a pulmonologist working on lung stem cells, talking to some of my cystic fibrosis experts around here, Dr. Wine and Dr. Mila, Dr. Kornfeld, and Dr. Moss. We put together a team to say, let's think about how we can apply genome editing to cystic fibrosis. And what we've started to do now is to say, well, what do we need to do? Instead of trying to solve the problem all at once, can we break it down into steps, each step of which we might be able to solve? And so the first thing we've done is said, well, we're not going to try to address all of the problems that cystic fibrosis patients have. Instead, we're going to start with one problem. And the problem that we've chosen to start with is the problem of the sinuses. So nearly all, or if not all, patients with cystic fibrosis end up developing sinusitis or congestion in their sinus. And it's not a disease, it's not a problem that patients necessarily die from, but it certainly impacts on the quality of their life and probably does contribute to their lung disease as well, because the infections in the sinuses can drain down into the lungs. And what we thought we would do, but the advantage of the sinuses is that they're very accessible, and our team member, Dr. Nayak, who's a ENT surgeon and regularly does sinus surgeries on patients with cystic fibrosis, so goes in and drains the sinuses of the pus and the infections, said that he can actually harvest the stem cells of the sinuses. So maybe what we should do, our first try should be to take stem cells from the sinuses, bring them into our laboratory, use our genome editing technology that we've gotten better and better at through diseases like sickle cell anemia, apply it to the sinus stem cells, and then figure out a way of giving those stem cells back to the sinuses. So we might not cure the lung disease right away, but we might be able to cure or significantly improve the sinus disease. And so we've gotten a grant from the California Institute of Regenerative Medicine, or CIRM, to support this team, and which is, as I said, a very exciting team because it's not just my lab, it's combining the expertise from a lot of other labs to do this. So where are we? Well, where we are is we get, Dr. Nayak, he gets the stem cells from the sinuses, and they go to his lab and Dr. Kuo's lab, where they have figured out ways of taking the small number of sinus stem cells, which are probably very much like the airway stem cells in the lungs, and expand them. So now we can grow them in the laboratory. So we can create a large number of stem cells that we can start with. And then the cells come to my lab, and we can use our genome editing technology, and we're able to modify the stem cells and correct the CF gene in right now around 10 to 20% of the cells we can correct the gene. We think that that's a really good starting point, but we think that we probably need to get that number up to 30, 40%, or figure out a way of taking the 10% of cells that are corrected and purify them so that they're up to 90% corrected. So I think we have a good start, and I think we have what we've learned from our sickle cell work is that when we're at 10 or 20%, often 6 to 9 months later, we're up to that 60, 90% level. So I'm very optimistic that we'll get there. So one of the differences, in addition to the blood being different than the lung, and there being different genes involved in sickle cell anemia versus cystic fibrosis, is that in sickle cell anemia, all the patients carry exactly the same genetic change, the exact same change in the DNA of their gene. So that means that our genome editing approach, we don't have to think about how to design a system for every patient because every patient has the same change. As many of you probably know, in cystic fibrosis, that's not the same situation. Now, we know that 70% of patients have at least the delta F508. So one of our genome editing approaches is to say, let's focus on that 70% that have the delta F508 and find a gene correction system for that specific mutation. And so we're pursuing that track. But what we don't want to do is say, well, does that mean genome editing is going to be left out for the patients who don't have that mutation? And our answer is no. And in fact, we work on other genetic diseases of the blood and immune system that are much more like cystic fibrosis, in which the mutations are spread throughout the gene. There's not one single mutation to correct. And so the strategy that we're adopting, and we'll see if this works, I think it'll work, because it's worked for some other genes, is to take what we call the cDNA, which is the whole gene, let's see, what's the way to describe a cDNA? It's the gene that just contains the part that will make the protein that is expressed on the cell. So that sequence, only that sequence, and we make that direct sequence, and instead of putting it somewhere in the cell or some random place, which is done in gene therapy, we're going to take that cDNA that has the fully functional CFTR gene and put it back into the CFTR gene right at the start. So what will happen is that the cell, when it's trying to make the CFTR gene, we'll make the good copy first, and then it will stop because we'll say, good, you've made the good copy, no need to read further. And so in this way, what we hope is that we'll have a universal system that will functionally correct all of the mutations that cause CF. One of the challenges is, can we efficiently get this full copy of the gene right at the start? As I said, we're achieving a frequency of about 10%, so we'd like to get that much higher. And then the second thing is to make sure that when we insert this full copy, that it actually rescues the cells, that we rescue the salt conduction problems that are present in cells. And so for that, we're collaborating, again, with our great group of faculty here, including Dr. Wine, to show that once we do this modification, we actually do correct the cells. So we have a Delta F508 specific system and then a universal system as well. After then correcting the stem cells, the next thing we'll need to do is think about how do we give those cells back to the patient. Early on, we thought, well, what we might do is just take the cells and sort of squirt them into the sinuses and hope that they go where they're supposed to do. But I think we all sort of felt like that might be a little bit too risky, or not risky to the patient, but it might not work. And so what we've decided to go down a path is to collaborate with engineers or bioengineers and take our corrected stem cells and embed them into what we call a matrix. So now they're on a solid scaffold. And so now, instead of just squirting the stem cells back into the sinus, what we've done, and again, Dr. Nayak's lab has been leading this, is to take the cells, embed them into a matrix, which then one day he will then be able to have another surgery where he goes in and not just squirts the cells, but lays the cells onto the lining of the sinus so the cells can then move from the matrix onto the tissue of the patient. We have shown that our cells can embed into this matrix, and we need, so what do we need to do to move this forward? Well, as I mentioned, we need to get our gene correction frequency a little bit higher, and then we need to show that the cells are not going to be dangerous. I have very much confident that they won't be, but before we start a clinical trial, we have to do some formal testing of that. And we have to show that when we put the cells into the matrix and we put the matrix into an animal, that the cells will do what they're supposed to do. And so we'll have to start doing some animal experiments before we will be able to try this in humans. And we hope that with the support of CIRM and some other funding agencies, including the CFRI and the CFF, that we will be able to accomplish these goals optimistically, maybe in 6 months, maybe more realistically in 12 to 18 months. When we have this, when we have this laboratory process developed and confirmed, of course, as people at Stanford University, we will publish it and we'll report on it so that we describe what we've done so other people can both tell us what we should do better and also so other people can build on it. But then we'll also need to then talk to the FDA, the Food and Drug Administration, about the process we've developed and propose to them how we intend on translating this into a clinical trial for patients. And so we expect that we'll be having our first discussion with the FDA probably in 2018 so that we can begin to come to an agreement on a roadmap for how we might take this strategy to a trial for patients. I can't say when it might be first available in patients, but of course everyone here is working hard to hopefully have it happen in the next couple years. So for those of you who have cystic fibrosis or are familiar with the disease because a family member has it or because you've become interested in it, you know that the sinus is not the most important organ that's affected. The most important organ is the lung, although of course the gut and pancreas and other organs are important as well. So how are we thinking about this in terms of, well, why are you just doing the sinuses? Well, we feel like the sinuses, which are connected to the airways, are our way of working our way down. So we feel like if this process works in the sinus, we will learn how to make the process better, so that then we can work down into the upper airways and then deeper into the lungs, so that we learn from each step in the airway where it's easier to get the cells and safer to give them back without having to go all the way into the depths of the lung, where it might be very challenging if we started there right away. So that's our strategy. I wish we felt like we could go right to the deep parts of the lung right away, but I think when I was in training, I heard a saying from a scientist who won the Nobel Prize, and what he said was, slow is fast. And what he meant by that is that sometimes the fastest way to get to the point you want to go is not by going there directly, but instead taking a measured, prudent, what seemingly slow process and working your way towards your goal. And so instead of taking 2 steps forward and 3 steps back, that you just keep taking 1 step forward, 1 step forward, 1 step forward, so that eventually you get there. So we're excited about the team we have at Stanford. We're excited to be more engaged with the cystic fibrosis community, both the community at large and the research community around the world. And hopefully we'll have the chance to come and talk on this podcast and at some future date describing the progress we've made. So thank you very much, and again, thank you for having me on.